अभी शुरू कर रहा हूँ मैं। आज के लिए पढ़ाई का मामला, तो आज इसमें अभी पढ़ूंगा मैं पूरा बोटानी जो है 4 घंटा लूँगा फुल रिवाइज करने में। और रिवाइज फुल या 50% तो आज करना ही पड़ेगा। जो लिस्ट ऑफ टॉपिक सिलेबस में है मेरा, लिविंग वर्ल्ड है, प्लांट किंगडम, प्रिंसिपल ऑफ इनहेरिटेंस और मॉर्फोलॉजी। 4 चैप्टर आज, फिर 4 चैप्टर कल। तो चलिए शुरू करते हैं। पहले हम लोग प्रिंसिपल ऑफ इनहेरिटेंस से शुरू करेंगे। लेट्स स्टार्ट। अरे तो आज जो चैप्टर्स हैं, लिस्ट ऑफ टॉपिक्स, वो है प्रिंसिपल ऑफ इनहेरिटेंस, नंबर 2 is morphology, number 3 is plant kingdom, number 4 is living world. This is the 4th chapter. Okay, so let's start it. Yes, we have same name. Genetics equals to inheritance. Explain how characters are passed on the progeny from parent to progeny, plus variation, degree by which progeny differs from their parents. Genetical terms, genes or factors, they are the units of inheritance which contain the information that is required to express a particular character in an organism. They are the units of inheritance. Alleles, genes which code for a pair of contrasting traits and present on the same locus, homologous chromosome, are known as alleles. They are the slightly different forms of the same gene. They are the slightly different forms of the same gene. Homozygous pure identical pair of alleles, TT, big TT or small TT. Heterozygous hybrid dissimilar pair of alleles, big T, small t. Phenotype, external and morphological characters. Homozygous identical pair of alleles, either it is homozygous pair identical pair of alleles, either it is TT or small t, small t. Heterozygous dissimilar pair of alleles, that is either big T, small t or big T, small t. Phenotype, external and morphological appearance of the character. Phenotype is external or morphological appearance of the character, and genotype is genetic makeup of an organism. Genetic is genetic makeup. Punnett square, it is a graphical representation to calculate the probability of all the genotypes of offspring in genetic cross. Experiments performed by Mendel on genetics and description of mechanism of heredity, process and formulation of principles are known as Mendelism. What is Mendelism? Experiments performed by Mendel on genetics and description of mechanism of heredity, process and formulation of principle are known as Mendelism. He conducted hybridization experiments on garden and garden pea Pisum sativum, that is Pisum sativum, and for 7 years, 1856 to 1863, and proposed laws of inheritance. Mendel conducted artificial pollination or cross-pollination experiment using several true breeding pea lines. A true breeding pea line is one that have undergone continuous self-pollination for several generations. Mendel selected 14 true breeding peas plant varieties as pairs which were similar except for one character with contrasting traits. Characters are stem height, flower color, flower position, pod shape, pod color, seed shape and seed color. So stem height, flower color, flower position, pod shape, stem height, flower color, flower position, pod shape, pod color, seed shape, seed color. Stem height, flower, pod, seed. Flower color, flower position, pod shape, pod color, seed shape and seed color. So stem height was tall and dark, flower color was violet and white, flower position axial and terminal, pod shape inflated and constricted, inflated and constricted, pod color green, yellow, seed shape round, wrinkled, yellow, green. So stem height, tall dark, flower color violet white, flower position axial terminal, cone shape, inflated constricted, pod color green or yellow, seed shape round or wrinkle, seed color yellow or green. So that's it. Reasons for Mendel's success. These experiments had a large sampling size. These have a greater credibility to the data that he collected. He concentrated only on one character at a time. He uses statistical analysis and mathematical logic for analyzing the result. The characters selected by Mendel were present on different chromosomes. His interference are confirmed from experiment on successive generation of his test plants. Okay? So these are the reasons why Mendel got success. Interference are confirmed from experiment on successive generation. Rohit Gamer. Hi. Okay, so ये था reasons for Mendel's success. His experiments had a large sampling size. This gave greater credibility to the data that he collected. He concentrated only on one character at a time. He used statistical analysis and mathematical logic for analyzing the result. The characters selected by Mendel were present on different chromosomes, and his interference are confirmed from experiment on successive generation of his test plant. Okay. Inheritance of one gene. Inheritance of one character at a time in an organism is called monohybrid cross. Okay, study of inheritance of one character at a time in an organism is called monohybrid cross. Conclusion of monohybrid cross. What was the conclusion of monohybrid cross? Law of dominance. Characters are controlled by discrete units called factors. Characters are controlled by discrete units called factors. Factors occurs in pairs. In a dissimilar pair of factor, one member of the pair dominates, the other is recessive. The dominant allele is a wild type or unmodified allele which produces normal সকালে 2 ঘন্টা নষ্ট করেছি আর কি পড়িনি So, study of inheritance of one character at a time in an organism is called monohybrid cross. Study of inheritance of one character at a time is called monohybrid cross. Phenotype Mendelian ratio 3:1 and genotype 1:2:1. Law of dominance. Law of dominance. Conclusion of monohybrid cross. Characters are controlled by discrete units called factors. Characters are controlled by discrete units called factors. Factors occurs in pairs. Factors occurs in pairs. In a dissimilar pair of factor, one member of the pair dominates and the other recessive. A dominant allele is a wild type or unmodified allele which produces normal enzyme protein that forms a character. A modified allele is a mutant allele which could be responsible for the production of less efficient enzyme or non-functional enzyme or no enzyme at all. Thus, it will be a recessive allele. So, in this law of dominance, there are 5 points. Characters are controlled by discrete unit called factors. Factors occurs in pairs in a dissimilar pair of factor. One member of the pair dominates and the other is recessive. A dominant allele is a wild type. Oh shit. A dominant allele is a wild type or unmodified allele which produces normal enzyme protein that forms a character. A modified allele is a mutant allele which could be responsible for the production of less efficient enzyme or non-function of enzyme or no enzyme at all. Thus, it will be a recessive allele. Law of segregation, law of purity of gametes. This law is based on the fact that the alleles do not show any blending, that both the characters are recovered as such in F2 generation. Though the parents contain two alleles during gamete formation, the factors or alleles of a P segregate from each other such that gamete receives only one of the two factors, thus gametes are always pure. The 3:1 ratio obtained in F2 generation is mainly due to segregation, although there is the role of the dominance also. So in this law, it is stated that no either the alleles completely blend with each other. They are like hidden in their parent. Likewise, in F2 generation, they showed the character. Deviations from Mendel's law. The laws are the deviation laws from Mendel's law are those laws that do not agree with the Mendel. Okay, so incomplete dominance. Incomplete dominance was discovered by Carl Corens, 1903 in Mirabilis Jalapa. When a cross between two pure breed is done for one contrasting character, the F1 hybrid phenotype does not resemble either of the two parents and was in the between the two, called incomplete dominance. So in this, the law do not resemble one of the parent, and it was resembling the complete different parent from the parent. Example, if a 4 o'clock or snapdragon plant with red flower is crossed with a white flowered plant, the offspring will have pink flowers. The offspring will have pink flowers because some red pigment is produced in F1 intermediate color plant. Neither the red nor white flower color is dominant. Such a situation is known as incomplete or partial dominance. Okay, such a situation is known as incomplete or a partial dominance. F2 generation phenotypic ratio is 1 is to 2 is to 1, means 1 is red color, 2 is pink and 1 is white color instead of 3 is to 1 as monohybrid Mendelian cross. Genotypic ratio of F2 generation is 1 is to 2 is to 1 and phenotypic ratio is also 1 is to 2 is to 1. So in inheritance phenotypic ratio is 3 is to 1 and in genotypic ratio Mendelian it is 1 is to 2 is to 1, means 1 tall, 2 big T small t and 1 is dark. Okay, so this is also genotypic ratio of complete dominance is 1 is to 2 is to 1. Tay-Sachs disease is an example of the result of incomplete dominance. Next is codominance. Tay-Sachs disease is the example. Incomplete dominance is also found in color of the flower in snapdragon or anthurium or dog flower and feather color in Aldulatian fowls. Now it is codominance. In codominance, the alleles are able to express themselves independently in F1 generation. Best example of codominance is the ABO blood grouping in human. Best example of codominance is the ABO blood grouping in human. Okay. In codominance, the alleles are able to express themselves independently in F1 generation. So, alleles are able to express themselves F1 generation itself. Best example of codominance in the ABO blood grouping in human. ABO blood grouping is controlled by gene I. The plasma membrane of the RBC has sugar polymers that protrude from its surface and the kind of sugar is controlled by the gene I. The gene I has 3 alleles, that IA, IB and small I. Each person possesses any 2 of the 3 I gene alleles. IA and IB are completely dominant over small I. Next is the alleles IA and IB produce a slightly different form of sugar while allele small I does not produce any sugar. When IA and IB are present together, they both express their own types of sugar. This is because of codominance, and red blood cells have both A and B type sugar, showing the genetic basis of blood group IA, genotype of offspring IA, IB, AB blood grouping. IA and small I, A blood grouping. IA, AB blood grouping. IB, B blood grouping. IB small I, B blood group. Small I, O blood group. Next is multiple alleles. Existence of more than 2 alleles of a gene. Since an individual 2 allele can be present, multiple alleles can be found only in which population studies are made. Number of possible genotypes n plus 1 divided by 2. Blood group inheritance in human is 3 alleles. So blood group inheritance is 3 alleles. And remember the formula that it is n plus 1 divided by 2. Next is back cross. It is a cross of F1 individual with any of their parent. Outcross F1 individual into homozygous dominant parent. Phenotypic ratio. Back cross. It is a cross of F1 individual. It is a cross of F1 individual with any of their parent. With any of their parent. outcross, and that is F1 individual into homozygous dominant parent, phenotypic ratio all dominant, genotypic ratio 1:1 for monohybrid cross. Test cross F1 individual into homozygous recessive parent. It is used for F1 individual, phenotypic ratio 1:1, genotypic ratio 1:1. Reciprocal cross, a cross in which male and female sexes are reversed, and in Mendelian cross, result of reciprocal crosses were same. So in reciprocal cross, a pair of crosses in which male and female sexes are reversed, and in Mendelian cross, results of reciprocal crosses were same. So pair of cross in which male and female sexes are reversed is reciprocal. Pair of crosses in which male and female sexes are reversed in Mendelian cross, result of reciprocal crosses were same. Okay. So here we ended our first session till our inheritance of one gene. Okay. Now it's a break time.
बचे हुए डेज नीट के अभी जो 4 मई का पेपर उसमें कुछ है
For many years, prudery or false modesty has caused the basic facts of sex to be withheld from large numbers of our young people.
It's break time, and we are coming back with 2 minutes. लैब बनेगी तो। 1 लाख कंप्लीट हो गया था। अब हम लोग पढ़ेंगे इनहेरिटेंस ऑफ 2 जीन। शुरू करते हैं। Dihybrid cross is a cross between two organisms of a species that are different in two pairs of contrasting characters. Mendel wanted to observe the effect of one pair of heterozygous on other pair, so he selected traits for dihybrid cross for his experiments as follows: color of cotyledons yellow green, color seed shape round wrinkled. Color of cotyledon color of cotyledon color of cotyledon yellow or green, and seed shape is round wrinkled. All the plants in F1 generation were yellow and round seeds. All the plants F1 generation were yellow and round seed. All the plants of F1 one generation is yellow and round. These factors, that means this. The factors of both character will independently segregate to each other during the gamete formation, thus total 4 types. Okay. YR, 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 okay, form in F1 generation. They segregate. In F1 generation, they segregate. The Punnett square given in the diagram can be effectively used to understand the independent segregation of the pair. The Punnett square the factors of both characters will independently segregate. The Punnett square diagram can be effectively used to understand the independent segregation of the two pairs of genes during meiosis. Law of independent assortment: when two characters dihybrid are combined in a hybrid, segregation of one pair of traits are independent of both the pair of traits. The above dihybrid cross between homozygous dominant smooth and yellow seeded with homozygous wrinkled and green provide this law. Prove this law. In test cross phenotypes and genotypes when two characters dihybrid are combined, what is independent law of assortment? Gemini koi. What is independent law of assortment?
The independent law of assortment, also known as Mendel's second law, is a fundamental principle of genetics that states, during gamete formation, the alleles of two or more different genes is sought independently of each other. In simpler terms, the inheritance of one trait does not affect the inheritance of another trait. The alleles for different traits get sorted into gametes randomly and independently. Here's a breakdown of what this means. Genes on different chromosomes. This law primarily applies to genes that are located on different non-homologous chromosomes.
During meiosis 1, when homologous chromosomes separate, the orientation of each homologous pair is random. This random orientation leads to different combinations of maternal and paternal chromosomes being packed into gametes. As genes are located on this chromosome, the alleles for those genes are also assorted independently. Genes far apart on the same chromosome, while genes are located on the same chromosome, are technically linked. If they are far enough apart, crossing over during meiosis can occur frequently. Dihybrid cross, example, consider pea plant. Explain this to a 15-year-old boy.
Okay, imagine you have two different sets of cards. Set 1, seed color. You have a yellow card, let's say Y, and a green card, Y. These represent the different versions, alleles, for seed color in a pea plant. Set 2, seed shape. You also have a round card, R, and a wrinkled card, R, representing the versions for seed shape. Now imagine a parent pea plant has one of
In a test cross, phenotypes and genotypes ratio are same. Non-allelic interaction. Epistasis: when a gene prevents the expression of another non-allelic gene, then it is known as epistatic gene. Phenotypic ratio: dominant epistasis 12 is to 3 is to 1. Example: hair color in dogs, fruit color in squash. Recessive epistasis: phenotypic ratio 9 is to 3 is to 4. Example: coat color in mice. When a gene prevents expression of another non-allelic gene. Complementary gene, phenotypic ratio 9 is to 7. Example: flower color in lathyrus. Supplementary gene, phenotypic ratio 9 is to 3 is to 4. 9 is to 3 is to 4. Coat color in mice. Important formulae applicable in case of selfing only. 2n types of gamete or types of phenotypes. 3n types of genotypes. 4n total number of possible zygote. So this is the formula. Epistasis: when a gene prevents the expression of another non-allelic gene. Epistasis: when a gene When a gene prevents the expression of another non-allelic gene, then it is known as epistatic gene, and this phenomenon is known as dominant epistasis 12:3:1. Who experimentally verified the chromosomal theory of inheritance? T.H. Morgan. What does the chromosomal theory of inheritance state? Genes are located on chromosomes and segregate independently during gamete formation. What does chromosomal theory of inheritance state? Genes are located on chromosomes and segregate independently during gamete formation. Genes are located on chromosome. What is linkage? The physical association of genes on a chromosome. What is recombination? Generation of non-parental gene combinations. What did Morgan experiment show about linkage strength? What did Morgan's experiment show about linkage strength? It is directly proportional to the. Okay, breath.
কিছু বেশি বলে
করছি আজ বলেছি
ইজি বানাবে এইটা তো কাজই করে না সিমেন্টটা বলে কোনো জিনিস না
95% ছেলেরা বিএসসি বিয়ে করে ভালো ভালো গোল্ড মেডেল করে বসে রয়েছে
আমি কোনো মতে
তারপর বলবে যে কেন 10,000 টাকা কামাস ও বলে 40,000 টাকা কামায় কি করছিস অফিসের চাকরির ওই কাজ করতে চাই চাপ পেতে চাই কাজ
যাবো একটা ইনস্টাগ্রাম পোস্ট দেখলাম
ওই যে মেনে নিলি না ভিআইপি আর বিটেক বায়োটেক এবার ভাবছি আরো কিছু করি না
সায়েন্সে বায়োটেকটা ভালো ছিল না বায়োটেক না সব
বায়োটেকটা সব জায়গা মানে একটু একটু ডিফারেন্স সেক্টর অনেক ঢুকবে না আর স্কিল থাকবে ব্যাপারটা এই দুটো জিনিস সেক্টর ঢুকবে কিন্তু হয়তো ওই কিরকম
আমি মনে করছি মনটা করে বলবো কাউকে খুলে দিয়েছি পাঁচ নয় বা ইউটিউবারটা একটু বেশি আমি পাঁচ
এখানে যে বিস্কুট ছিল ভালো তো নাকি ওই তো কিনে দিতাম
রুটি তারপর
আমারও বাইরে কি পছন্দ ভালো লাগে ঠিক আছে আর তারপর কি রে কি হলো
নিয়ন
জানি সোজা নয় সোজা সোজা কঠিন আছে রাস্তা It's not like বাইরের বিদেশের ইন্ডিয়ান আমরা চাই যেতে থাকতে একজনই যেতে পারি
না
আমি কয়েকজনের মধ্যেই যাবো যেতে চাই চেষ্টাটা করো বাইরে চলে যাবো বিদেশ চলে গেলে না ভালো যত টাকারই যাবে হিউজ কোনো টাকা চাকরি করবো না তাও
ইনফর্ম করে আর মনে হয় না যেতে পারবি বলে মানে অপরচুনিটিটা থাকবে আর বিদেশ যাওয়ার শখ কেন
ভালো লাগে
চাকরি করার জন্য তাও ইনফর্মের পর লোক ইনফর্ম করতে গেলে ওখান থেকে করলে তবে না চাকরি পাওয়ার বেশি সম্ভাবনা এখান থেকে করে গেলে ছেড়ে দে সম্ভাবনা তারপর তারপর হয়তো এন্ড আপ করে যাবি কোনো পাউটির দোকানে কাজ করতে চাকরি পাবি না এখানে ডিগ্রি বলবে এই নাই সেই নেই যদিও পৌঁছে যাস তো দিনক্ষণ চালাবি আর চাকরি খোঁজার যে দিনগুলো থাকবে সেই সময় ওই পাউটির দোকানে হয়তো কাজ করতে হবে পাউটির দোকানেও বলছে কানাডাতে পাউটির দোকানে পিজা কোম্পানিতে চাকরি পাওয়ার জন্য 10 জনকে ডেকেছে চলে এসেছে 550 জন লাইনে দাঁড়িয়ে আছে স্টুডেন্টসরা জব সিকাররা হচ্ছে ইনসেন ভাবে ইন্ডিয়ানস ভরে গেছে নোংরা ভাবে সব চাইছে পার্ট টাইম জব কেউ চাকরি খোঁজে গেছে পাচ্ছে না আসল চাকরি মাঝে সময় লাগছে ইন্টারভিউ দিচ্ছে ততদিনে ভাবছে করি পিজা। জ্যানিটরের কাজে 10 জন রিক্রুটমেন্ট করবে না কত করবে বলে 5 জন করবে বলেছিল, পৌঁছে গেছে 55 জন। জ্যানিটর কানাডায়।
জানি বিদেশ তো যায় আমাদের জন্য।
এরম ছিল না 5 বছর 2 বছর 4 বছর আগে সিস্টেম এরম ছিল না। তুই পিজা হোমে চাকরি দিয়ে আসানিসে জব পেত লোক। তুই বিদেশ কথাটা মাথা থেকে হাঁটা।
না পারলে আমি বাইরে যাবো, আমার ওইটাই কারণ। আমার কোনো কথা
বায়োটেকে তো আরো ডুবে যাবি, এখানে আরো ডুবে যাবি বায়োটেকে। ফার্স্ট ইয়ার থেকে ডুববি।
বেশি পাবো।
মাইনে বেশি পাবি কি, এমফার্ম করলে এখানে একই তো সমতুল্য মাইনে পাবি।
তাও না, তাও কিছু
কত মাইনে, ওটা তো লেখা থাকে, লেখার উপর ভরসা করে এগোয় কেউ?
ভরসা করে বলছি।
অতটাও ভরসা করতে নেই, ওটা একটু 2-4 গুণ বেড়েই যাবে, ততদিনে সব ঠিকঠাক হয়ে যাবে। লোক এমফার্ম করে ড্যাম কামাবে এখানে। বিদেশ কথা মাথায় থাকলে ড্রিম শ্যাটার হতে পারার চান্স আছে খুব বেশি। ভালো চাকরি করবি কি করে, স্কিলটা বাড়াবি কি করে, ওটায় ফোকাস কর। আর সেই স্টেজে নেই যে তোর ফ্যান্টাসি ওয়ার্ল্ডে তুই থাকবি তাহলে হবে। ফ্যান্টাসি ওয়ার্ল্ড করার জন্য আমাদের 2019-এ ভর্তি হয়ে যেতে হতো, মানে আমাকে বা তোকেও 12-এর সাথে সাথে, তবে ফ্যান্টাসি করার ওই তুই কামলি ভাবতে পারবি। না আছে এখন স্কিল, না আছে ডিগ্রি। আবার বলছিস তো নিট দিবি, তাহলে তোকে আরো খাটতে হবে। এখন তো ওইগুলো মাথায় নিয়ে চল। প্রায় এমবিবিএস এর মতো লং টার্ম, তার চেয়েও বেশি জার্নি। এখন থেকে বিদেশ ভাবলে চলে? ইচ্ছে রাখ, মনের কোণে ফেলে রাখ, ওইটার জন্যই একদম যাচ্ছিস, তাহলে তো বিশাল বলবো ব্লান্ডার মিস্টেক করতে চলেছিস আবার। তাহলে যেটা ভালো লাগে কর। যদি নিট ফিট সত্যি দিতে চাস, বিফার্ম পড়ে তাহলে লেগে পড়তে হবে। এমফার্ম করার পর, এখন এমফার্মের তো কথা ভাবিস না, ফার্স্ট ভাব বিফার্মে আমি কি করে সুন্দর করবো সব কিছু। বিফার্ম করার পর বিদেশটা ভাব। এমফার্ম করতে ওখানে যেতে হয় লোকে, তবে তো দেখ একটা তোকে দেশ কেন চাকরি দেবে। দেখ এদিকে দেখ, মাথায় এটা ভালো করে খাটা। ধর একটা পাইলট এইখানে পড়লো, বিদেশে গিয়ে বলছে আমি
সারাক্ষণ অক্টোগাইজ বলে ফিল করানো বন্ধ করে।
Dihybrid cross is a cross between two organisms of a species that are different into two pairs of contrasting characters. Mendel wanted to observe the effect of one pair of heterozygous on the other pair, so he selected traits of dihybrid cross for his experiment as follows. Color of cotyledons yellow and green, seed shape round and wrinkled. Yellow and round characters are dominant and green and wrinkled seeds are recessive characters. All the plants in F1 generation were yellow and round seeds. The factors of both characters will have independently segregate to each other. তুই ড্যাডিকে এটা বোঝাবি যদি বেশি বলতে আসে বি ফার্ম করলে আমার কাছে সুযোগ থাকবে আমি স্কুল টিচারেরও চাকরি করতে পারবো আর কোম্পানি জব সেটা আমার চয়েস
কমে যাচ্ছে
কি সুযোগ কে বললো বিটেকরাও তো করে
বেশিরভাগই এই নর্মাল লাইনই শুনতে পাই ও কাকুর আগে শুনতিস
তুই না না শুনিনি নয় মেন টিচার হতে গেলে কি লাগে গ্র্যাজুয়েশন ডিগ্রি
বিয়ে
গ্র্যাজুয়েশন ডিগ্রি প্রাইভেটে আর বিএড হলে ভালো হয় প্রাইভেট সেক্টরের কথা বলছি তো সেখানে তুই তো বায়োলজির নলেজ যদি প্রডিউস করিস তো সেখানে পড়ানো যাবে ওটা এমন নয় যে তোকে আবার বায়োলজি বিএসসি করতে চলে যেতে হবে তাহলে এত লোক কি সবাই বায়োলজি বিএসসি করে আছে নাকি কেউ কি বিকম করেনি
যখন যত জয়েন করে গেছি তাই সেম
হ্যাঁ তো তাদের কথা ছাড় না
তাদের তো দেখছে
তাহলে যারা ফিজিক্স কেমিস্ট্রি করে তারা বিটেক করে আসছে কেন এই নতুন কোর্স ওটা 10-15-20 বছরের আর সেরকম ও তো আসে না টিচারই লাইনে আলাদা আচ্ছা বিফার্মে টিচিং হবে না এমফার্ম করে লোক প্রফেসরই হয়ে যায় শুনিস নি আলাদা কথা কিন্তু হয় সবই সম্ভব বিফার্ম করে বায়োলজির নলেজ ইন্টারভিউ নেবে তো তোকে কি দেখবে তোমার ডিগ্রি নেই তোকে তো ইন্টারভিউ নেবে প্রাইভেট জানবি সব সময় স্কিলের ওপর চলে ওটা
বসে আছে
হ্যাঁ
বসে আছে
হ্যাঁ স্কিল ওটা তো জাজ করবে না কিসের ডিগ্রি হ্যাঁ এবার তুই ইংলিশের ডিগ্রি নিয়ে যদি তুই বায়ো পড়াবো ভালো কথা তো 12 পাস কেন দিয়েছে ওকে 9-10 পড়াতে 11-12 পড়িয়েছে নলেজ নিয়েছে টেস্ট নলেজের টেস্ট নিল হ্যাঁ তোরে তুই মেন ফোকাস তো তুই কন্টেন্ট দিতে পারবি কিনা তুই ডিগ্রি নিয়ে নিবি কন্টেন্ট জিরো তুই কোথাও চান্স নেই প্রপোজড স্যার ফিজিক্সটা দেয় যেমন ভরপুর দেয় আলাপ স্যার তুই চাওয়ার চেয়েও বেশি দেবে ওই জন্যই স্যার জিতে গেছে এই একটা ফিজিক্সের বদলাচ্ছে proposed by Walter and Sutton Theodore Boveri. The experimental verification of chromosomal theory inheritance was done by T.H. Morgan. Okay, linkage and recombination. Inheritance of gene in group is called linkage. The term linkage was coined by T.H. Morgan. Linked genes are located on the same chromosome. Linked genes are located on the same chromosome and all the আমার লাইভটা খুলে দেখ linked genes are located on the same chromosome and all the genes located on one pair of homologous chromosomes form one linkage group. In linkage, the proportion of parental gene combinations are much higher than non-parental type. Tightly linked genes shows very low recombination while loose linked gene shows higher recombination. Linkage may be complete, crossover, absent or incomplete, present. Percentage recombination equals to number of recombinant organism by total organism into 100. 1% CO is 1 centimorgan.
Oi.
हाँ
Physics ওয়ালা ফেরত অনুষ্কা ম্যাম এখন ফিজিক্স ওয়ালার টিচারের স্টুডেন্টের চ্যানেলে পড়াচ্ছে
কে
রক্ষিতা সিং এর চ্যানেলে
ফিজিক্স वाला কে বললো ফিজিক্স वाला স্টুডেন্ট রাক্ষিতা সিং
ফিজিক্স ওয়ালার টিচারের পার্সোনাল স্টুডেন্টের চ্যানেল অমিত
ফিজিক্স
স্যারের
वाला
স্টুডেন্ট ছিল
টিচারের পার্সোনাল স্টুডেন্ট
হ্যাঁ রক্ষিতা অমিত স্যারের স্টুডেন্ট ছিল
কবে জানলি এটা
বাহ অমিত স্যার ভিডিও কল রক্ষিতার বলেছে আমার নিজের স্টুডেন্ট রক্ষিতা
কোন ইনস্টিটিউটে
সেটা জানি না आज वो इतना अच्छी कर रही है
সেটা তাহলে বোধহয় ইনস্টিটিউটেই কোনো ইনস্টিটিউটেই সেটা কি ইনস্টিটিউট বলতো অ্যালেন রক্ষিতা সিং অ্যালেনে পড়েছে
ইসলাম আমি যে ফিজিক্স ওয়ালার স্পেশাল টিচারের নিজস্ব স্টুডেন্ট
নিজস্ব মানে
এখন চলে গেছে নিষ্ঠ ম্যাম ভুল ভাল অঙ্ক করাতে সরি বেটা সরি 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 কি রিপিটিং ভালো লাগে না আমার
হেড করছিস কেন
কি
হেড করছিস কেন
কারণ অনেক জ্বালিয়েছে লিখে নিতাম পুরো কোশ্চেনটা পুরো কাটতে হতো ওর জন্য আবার মুছে লিখতে হতো
হেড করছিস কেন
ভালো বই লিখতে পারি না এই
percentage recombination or recombinant frequency or directly proportional to distance between gene. Percentage recombination क्या होता है नंबर ऑफ recombinant organizing percentage recombination क्या होता है number of recombinant organism डिवाइडेड बाई टोटल ऑर्गेनाइजिंग इनटू 100 ठीक है इसमें से कुछ सम्स है जो मैं आपको दिखाना चाहूँगा sex determination in honey bee female they develop from fertilized egg having two sets of chromosomes from both parents genetic basis okay principle of inheritance in a dihybrid cross 20 recombinant percentage recombination number of total organism 20 recombinant organisms are found in a total of 200 offspring calculate the recombinant percentage recombination is given as we have to find number of recombinant organism is 20 number of total organism is 200 percentage recombination is 20 divided by 200 into 100 equals to 10%. If recombination frequency equals to 16%, total progeny is 500. Total progeny means total organism. Find the number of recombinant organism. So percentage recombination is given as 16%. Total progeny is given as 500. Total organism. Find the recombinant organism. Linked genes are located on the same chromosome and all the genes located on one pair of homologous chromosomes from one linkage group. Okay, in linkage the portion of parental gene combinations are much higher than non-parental type. Tightly linked genes shows very low recombination. तो अगर एक जीन में दो अगर एक आई कलर है ब्राउन कलर और उसका जो नोज कलर है मतलब सॉरी उसका जो हेयर कलर है ब्राउन, वो पेरेंटल जीन है तो वो ज़्यादा शो होगा, चांसेस है क्योंकि वो टाइटली लिंक्ड है, वो सेपरेट नहीं होते, तो हाई चांस है बच्चे में भी सेम होगा। तो लिंकेज डिस्टेंस बिटवीन जीन्स इज डायरेक्टली, रिकॉम्बिनेशन फ्रीक्वेंसी इज डायरेक्टली प्रोपोर्शनल टू डिस्टेंस बिटवीन जीन। तो टोटल ऑर्गेनाइजिंग 500% दिया है और पूछ रहा है टोटल फाइंड द नंबर ऑफ रिकॉम्बिनेंट ऑर्गेनाइजिम। तो यहाँ पर 16 इक्वल्स टू x बाई 500 इंटू 100, तो ये आपका हो जाएगा। फिर यहाँ पर और एक क्वेश्चन था, नहीं, इफ recombinant frequency is 1, recombinant another 45, what type of linkage is that? 1% is tightly linked, 45% is loosely linked. In a cross of 2000 pollination offspring of 10, recombinant frequency is 16%. How many will be parental types? Oh, in a cross 200 offspring of 10, तो क्वेश्चन बोला है 2000 ऑफस्प्रिंग ऑब्टेंड हुआ है, ठीक है। सो 2% रिकॉम्बिनेशन 2016% में 2000 रिकॉम्बिनेंट परसेंटेज में 16% और नंबर ऑफ हाउ मेनी विल बी, अच्छा, इन अ क्रॉस 2000 ऑफस्प्रिंग ऑफ 10, 2000, और अब क्या है इसे पूछ रहा है नंबर ऑफ रिकॉम्बिनेंट ऑर्गेनिजम्स, तो 16 इक्वल्स टू x बाई 2000 इंटू 100, इसमें x निकला 320 नंबर ऑफ रिकॉम्बिनेंट फ्रीक्वेंसी। अब 16% जो है पेरेंटल टाइप्स कितना हुआ, तो स्टेप टू कैलकुलेशन ऑफ टोटल नंबर ऑफ ऑफस्प्रिंग, टोटल नंबर ऑफ ऑफस्प्रिंग parentals equal to recombinant plus parental, total number of offspring equals to recombinants plus parentals, parentals equals to recombinant organism 2000 minus 320, so parental types है 1680, 1680 organisms are parental types. Oh, total offspring, formula is total offspring equals to recombinants plus parentals, recombinant निकला है प्लस पेरेंटल, पेरेंटल निकला है 2000, सो 2000 माइनस 320 इक्वल टू 1680। So here it is break. Now in the next labs we will be doing polygenic inheritance. Okay, polygenic inheritance, which will be done mutation, genetic disorder, pedigree analysis. In this lab we will complete uh, ম্যাথস ছিল তাই বিটেক করতে হবে। Okay, it's long break, 15 minutes. I will skip long break. Okay, shit. Sex determination. Let's start about sex determination. This type of sex determination, XX XY type, first observed by Wilson and Stevens in an insect. It is further categorized into two types, XX female and XY male. আচ্ছা, এটা হয়ে আছে। XXXY type. Okay. Let's start. Types of sex determination, XX and XY type. This type of sex determination first observed by Walter Wilson and Steven in an insect. It is further categorized into two types, XX and XY type. This type of sex determination first observed by Wilson and Stevens. This type of sex determination was first observed by Wilson and Steven. Wilson and Steven. XX female and XY male. In this type of sex determination, female is homogametic, produce only one type of gamete. Male is heterogametic, male produce two types of gamete. XY female. So Wilson and Steven find this type of sex determination was observed by Wilson and Steven in an insect. It is further categorized into two types. Further categorized into two types. One XX female and another is XY male. In this type of sex determination, female gametes are homogametic, produce only one type of gamete. Produce only one type of gamete. And XY female is heterogamete. And male produces two gamete, that is heterogametic. For male it is X and Y, and it is heterogametic. Okay. Next is XY female and XX male, or ZW female or ZZ male. So XY female and XX male. Ulto. XY female and XX male, or ZW female or ZZ male. ZW female or ZZ male. Produces two types of gametes, and male individual is homogametic, produce only one type of gamete. Sex determination, female is heterogametic, produce two types of gamete, and male individual is homogametic, produce only one type of gamete. It is found in some insects like butterflies, moths, and vertebrates like bird, fishes, and reptiles. It is found in some insects like butterflies, moths, and vertebrates like birds, fishes, and reptiles. XX female and XO male. In this type of sex determination, there is a deficiency of one chromosome in male. The female is homogametic and male is heterogametic. So, there are two types of sex determination. Example: grasshopper, cockroach, ascaris, and in plants, Diosceria santa and Vallisneria spiralis. Grasshopper, cockroach, ascaris. Okay, grasshopper, XX female and XO male. In this system, it is a XX female, so it is homogametic, and hetero also homogametic, and male is heterogametic. Okay, and homogametic, so one gamete is absent. One gamete is absent. And example is grasshopper, cockroach, ascaris for animals. Example: Dioscorea, sinuta, and Vallisneria spiralis for plants. Sex determination in humans. Sex chromosome 1 pair, XY male, female XY, and autosomes 22 pairs. X and Y sperms, X and X eggs. Female to male ratio is 1:1. Thus, there is an equal probability of fertilization of the ovum with sperm carrying either X or Y chromosome. Sex determination in honeybee. It is important. The sex determination in honeybee is based on the number of sets of chromosome an individual receive. An offspring formed from the union of a sperm and an egg develops as a female queen or worker, and an unfertilized egg develops as a male by means of parthenogenesis. This means that the males have half the number of chromosomes than that of a female. The sex determination in honeybee is based on the number of set of chromosomes an individual receives. And offspring formed on the union of sperm and an egg develops as a female formed from the union. Okay. From the union of a sperm and an egg develops as a female, which is known as queen or worker. and an unfertilized egg develops as male drone by means of parthenogenesis. This means that males have half the number of chromosomes than that of a female. So, by proper fertilization of eggs and sperms develops into female queen or worker. By an unfertilized egg into sperms develops into male. So there is 32 chromosome, and in male there is only 16 chromosome. Okay, the female are diploid having 32 chromosome and male are haploid having 16 chromosome. This is called haploid diploid sex determination system. So male drone haploid only 16 chromo, female queen worker diploid 32 chromo. This is known as, whole is known as 16 chromosome. A haploid diploid sex determination system. Haploid diploid sex determination system, and has a special characteristic features such as the male produce sperms by mitosis. They do not have father and does not have sons, but have a grandfather and can have grandsons. So the males produced by mitosis, they do not have father and thus no sperm. So by mitosis, male develops by insect, genotype of female XX, genotype of male XO, Drosophila and human XX, XY. Birds, fishes and butterflies, female WZ, male WW. So bird, fish and butterfly are the gametes, are type of birds, females, birds, fishes, reptiles are XY female and XX male. Means females are homogametic, heterogametic, and males are homo. And in Drosophila, XX female, XO male. If there is one gamete absent. Grasshopper, cockroach, Ascaris, and Disolcimenta Palissus spiralis. Okay, so it is done. Now next topic is topic number 9, that is mutation. Sudden inheritable change in an organism is called mutation. Mutation results in alternation of DNA sequence and consequently result in changes in the genotype and phenotype of an organism. Types of mutation: chromosomal mutation or chromosomal aberration. It is due to structural and numerical changes in chromosome. Structural changes of chromosomes involve following types: deletion, duplication. A part of chromosome is lost from the terminal deletion or intercalary path intercalary deletion. Example: deletion of a segment of short arm of fifth pair of chromosome in human causes cry-do-chat syndrome, cat cry syndrome in child. Deletion of a segment of short arm fifth pair of chromosome in human causes cry-do-chat syndrome. Duplication: it occurs due to addition of a part of a chromosome. Polyploidy can be artificially induced by application of colchicine and granosine. Colchicine and granosine. Polyploidy, colchicine and granosine. Polyploidy, polynixin and granosine. Gene mutation, sudden stable character. Fifth, deletion. Fifth pair of chromosome cry-do-chat syndrome. That's it. Gene mutation, chromosomal mutation or chromosomal aberration. Deletion, duplication. Gene mutation, sudden stable changes in structure of a gene or secretome due to change in nucleotide sequence and nucleotype are called gene mutation. Most of these genes include change in single nucleotide. This is called a point mutation. Sickle cell anemia is known as single nucleotide change. The gene mutation can be caused by deletion, insertion, frame shift, point. Deletion: one more nucleotides are eliminated from segment of DNA or gene. One more nucleotides are eliminated. Insertion: addition of one more nucleotides in the segment of a DNA gene. Frame shift: both deletion and insertion can lead to frame shift mutation. In this type, the entire reading frame of base sequence shift laterally either in the forward direction or in the backward direction. Either in the forward direction or in the backward direction due to deletion of one or more nucleotides. Due to deletion of one or more nucleotides. Point mutation. Mutation arises due to change in a single base pair of DNA. Mutation arises due to change in a single base pair DNA. Point mutation: the entire reading frame of base sequence due to insertion of one more nucleus or deletion. Entire reading frame the entire reading frame of base sequence shifts laterally either in Point mutation mutation arises due to change in single base pair. Point mutation change arises due to change in a single base pair or DNA. Frame shift mutation, entire reading frame of base sequence, entire reading frame of base sequence, shift laterally, shifts laterally, either in forward direction or backward direction. That's it. Deletion, one more nucleotides are eliminated from the segment DNA. Insertion, addition of one more nucleotide. Point frame shift is due to both deletion and insertion can lead to change in frame shift mutation. The entire reading frame is shifted laterally either in forward direction due to insertion, either in backward direction due to deletion. Point mutation arises due to change in single base pair DNA. Gene mutation, sudden stable change. Example, most of the gene include change in single nucleotide, that is sickle cell anemia. So mutation is completed. Induced mutation, they are produced by the external factors and chemicals. The first induced mutation produced by Muller 1927 in Drosophila through the use of X-ray. Mutagens, any type of physical and chemical factors that is used in the induction and mutation, is UV radiation and gamma radiation. They can change any type of physical or chemical factor. X-ray causes cancer. They are produced by the application of external factors and chemicals by Muller. Sudden stable change in the structure of a gene or cystron due to change structure of gene or cystron due to change in nucleotide sequence. Deletion, chromosomal mutation, one is gene mutation. Okay. So we completed the mutation part also. Now the disease part is left. We will continue this tomorrow, but 85% of the chapter is completed. Chromosomal theory of inheritance proposed by Walter and Sutton. Behavior of chromosome was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Gene occurs in pair. Chromosome occurs in pair. Segregate at the time of gamete formation such that only each pair is transmitted to a gamete. Segregate at gamete formation and only one each pair is transmitted to a gamete. Independent pairs, one pair segregates independently of the another pair. The experimental vacation chromosomal theory of inheritance was done by T.H. and Morgan, who gave Walter Sutton and Theodore Boveri, who experimented T.H. Morgan. Inheritance of many genes. Okay, inheritance of character in one is controlled by polygenic inheritance. Inheritance of character in one character is controlled by many genes. Intensity of character depends number of dominant allele. Dominant alleles are contributory in nature. Kernel color of wheat 2 gene ratio 1:4:6, 4:1. Skin color of human 3 genes ratio 1:15:15:20:15:16:1. 15:6:1 repeated. And 2 genes 1:4:6, 4:1. That's it. So it's break time and we have completed. Inheritance of one gene in a group is called linkage. The term linkage was coined by T.H. Morgan. Linked genes are located on same chromosome. Now we can do questions, but after break. Okay. Okay, so I'm stopping the timer and or I'm increasing it. Okay, I'm stopping it and I'm returning after few minutes. যাই। ঝামেলা করে ফেলি। হ্যালো।
আইসক্রিমটা দিবি না?
ও, যাই। চ্যাপ্টার হলো তাও আধা ঘন্টা পুরো তো হচ্ছেই না। ইংরাজিতে ছেড়ে দেওয়া খুব অবস্থা আছে। Okay, let's start. इरफान, तो चलिए शुरू करते हैं। अभी ब्रेक में 3 मिनट बाकी है, तो अभी हम लोग करेंगे प्रिंसिपल ऑफ इनहेरिटेंस का कुछ क्वेश्चंस। ठीक है, Mendel conducted hybridization experiments on a garden for Mendel proposed the law of inheritance in selection of Pisum sativum for hybridization experiment because of presence of 7 characters or short lifespan, presence of contrasting and short lifespan, presence of long lifespan and profile from many observed hybrids. Among the following given traits, Pisum sativum, which one is reversible trait? হ্যাঁ,
ধুলো ঢুকবে।
ও
আমি তো ফিরে দালাল পুকুর অব্দি গিয়ে হু করে যেই ঝড় উঠেছে আমি আর গাড়ি ঘুরিয়ে নিয়ে পালিয়ে এলি।
হ্যাঁ,
সবজি আনতে যাচ্ছে না, আমতলায় এখন এই স্কিমেতে দেয়
ও
নটার পরে। স্কিমে ঢুকে যাচ্ছে না। রাম ওই দালাল পুকুর অব্দি গেছে আর হু করে ঝড়টা উঠবে। এখান থেকে বেরিয়েছি একটু কি হাওয়াটা উঠছে, আমি যে হাওয়া দিচ্ছে
হেঁটে গেছিলে না টোটোয় যাচ্ছিলে, বাইকে গেছিলে?
হ্যাঁ, এখান থেকে যাচ্ছি ঠান্ডা ঠান্ডা হাওয়া। এবং মানসিকের বাড়ি যেই ক্রস করে আর একটু গেছি শিশিরের দোকানে দেয় সারা হু করে পাতাগুলো সব রাস্তার উঠতে আরম্ভ করেছে। তারপর আমি ওই মোড়টা গেলাম, ডাকগুলো মোড় থেকে ধরিয়ে নিয়ে পালিয়ে।
Okay, let's start. The time has started. Mendel has worked on Pisum sativum for 7 years by taking 14 true breeding location of traits on chromosome number. Okay. Which of the given gametophyte represent homozygous? Homozygous represent this. What is the degree by which progeny differ from their parent? Which part of the following trait of pea plant was not studied by Mendel in his experiment? What is the process of characters are passed from parent? A true breeding line is one that have undergone cell Select the incorrect statement. Inheritance is the process by which the characters are passed from parent to progeny. Inheritance is the basis of heredity. Variation is degree by which progeny differ from their parents. Sahiwal breeds of Burma Majha are produced through artificial and domestication of wild cows. Which of the following statement is incorrect regarding the experimental model of bicep sativum? It may undergo cross-pollination naturally. Tall plants are dominant over the green pods are recessive over yellow pods. Constricted pods are dominated over inflated. (52, 48). Accuracy 84%. Next TBD. A pea plant having violet colored flower with unknown genotype was crossed with a plant having white colored in the progeny. 50% of the plant were violet and white. The genotype considered the parent having violet colored flower was heterozygous. Law of purity of gamete was explained by Mendel and proved with the help of how many different types of gametes can be formed from one generation resulting from this. The individual with genotype will produce eight different types. A cell is heterozygous at gene loci. How many different types of gametes can it form? Alleles do not show any blending and both the characters recover as such in F2 generation. Factor of the discriminant on the particular factor. In monohybrid cross, pure homozygous plant in F2 generation will be Punnett square is law of dominance and law of segregation was based on cross between tall and dark pea plant. Kelo. Cross between রায় এলো রায়ের মুখখানা দেখি ও বাবা এ হলো রায় নরমাল নাগরিক ইউথ অফ ইন্ডিয়া দেখো ইউথ অফ ইন্ডিয়াকে চোখে স্বপ্ন আছে এই দেখিয়ে দিয়েছে
ভালো করে দেখ স্বপ্ন
হায় এবার তো কনফিউজ একে ভালো করে করো
পথ দেখাও
হ্যাঁ
cross between tall and dark plant to study the inheritance of
cross between tall and dark plants to study the inheritance of one gene na two gene tall and dark planter মধ্যে ক্রস করাচ্ছে to study the inheritance of
something I wanna see
হ্যাঁ both A and B
কেন বল
ওয়ান জিনেও করতে পারি, টু জিনেও করবো।
আমার আছে মনোহাইব্রিডে যেরকম
মনোহাইব্রিড আর ডাইহাইব্রিড।
ডাইহাইব্রিডেটাও
ডাইহাইব্রিডে
রাখবো
দুটো জিন রাখে না রে, কারণ
তার
এটা
মানে কোশ্চেনটা
মনোহাইব্রিড।
জিজ্ঞেস করতে চেয়েছে শুধুই টল অ্যান্ড ডোয়ার্ফ, মানে বেশি মাথা ঘামাতে তোমার লাগবে না, এইটা হচ্ছে মিটার একটা কারণ, বেশি ভাববে না, সিম্পল কোশ্চেনের
Tall
সিম্পল।
and dark tree plants. A test cross enables the determine distinguish between. Test cross হচ্ছে প্যারেন্টের সাথে। Determine the viability of cross. Distinguish between homozygous and dominant and heterozygous dominant. Determine whether two species can interbreed. Determine the similarity DNA to distinguish between homozygous dominant. হ্যাঁ, test cross. টেস্ট ক্রস,
কেন?
শুধু টেস্ট ক্রস বলেছে তাই দুটোই হবে। যদি বলতো ব্যাক ক্রস তাহলে হোমোজাইগাস ডমিন্যান্ট প্যারেন্টের সাথে করালে ব্যাক ক্রস। হ্যাঁ, আউট ক্রস হিটারোজাইগাস, ওরও হোমোজাইগাস ডমিন্যান্ট হোমোজাইগাস রিসেসিভ হয়। হিটারোজাইগাস ডমিন্যান্ট, হ্যাঁ হিটারোজাইগাস টেস্ট ক্রসের মধ্যে সবই পড়ে। টেস্ট ক্রসের যদি আবার আসে তাহলে আছে ব্যাক ক্রস আর আউট ক্রস। ব্যাক ক্রসটা জানি হোমোজাইগাস ডমিন্যান্ট, এটা আমি পড়িনি, এটা কোনেতে হয়। আর একটা পড়েছি হোমোজাইগাস রিসেসিভ, যেটা আউট ক্রস। বেসড অন দিস অবজারভেশন মেন্ডেল প্রপোজড, এইটা বোঝাতে গিয়ে সিট ম্যাম अगर मान लो टेस्ट क्रॉस मतलब तुम्हारा शादी अगर मम्मी के साथ, छोड़ो ऐसा नहीं, इंग्लिश में बोला जाता है। F2 प्रोजेनी में जो है उसको अगर F1 से कंबाइन करे तो क्या होगा? कंट्रोवर्सी हो जाता है। ह्यूमन ऐसा হয় একটা সায়েন্টিস্ট ছিল, করতে চেয়েছিল এরকম হিউম্যান, মানে যেমন মেন্ডেল পিককে নিয়ে করেছে ও হিউম্যানে করতে চেয়েছিল, এথিক্যালি বারণ করে দিয়েছে।
আমার সিস্টার হয়,
হ্যাঁ
স্যার সব থাকে বেশ আমাদের
এথিসিস্টরা খুব প্রচন্ড বিরোধ করেছিল। আর ও বলেছিল আর একটা যে হিউম্যানকে যদি আমি দেখতে চাই 10টা হিউম্যান বেবিকে পাইরাকে একটা রুমে রাখবো, তার কিরকম ডেভেলপমেন্ট হয় ব্রেনে, অ্যাজ আ আমাদের রুমে রাখবো না জঙ্গলে ছাড়বো একটা, মানে উইদাউট প্যারেন্ট জঙ্গলে ছাড়বে
ছোট বয়সে ছাড়বে,
একটু বড় করে কেয়ার নিয়ে একটু বড় করে যদি চলতে পারবে, মানে সিলেক্টেড, মানে ক্যামেরায় অলওয়েজ অবজারভেশন হবে, ওদের ফলমূল দিয়ে দেওয়া হবে আগে থেকে যাতে খায়, কিরকম ন্যাচারাল হ্যাবিটেটে বড় হয়, আপ টু 18 দেখবে বলেছিল। এটাও এথিক্যালি বাউন্ড করে দিয়েছে। তাদের এমআরআই করতে, যে তাদের কি ডেভেলপমেন্ট হচ্ছে ব্রেনে,
কি হলো না,
হ্যাঁ কিন্তু বলেছে না হিউম্যানে করতে।
মানুষের সাথে অন্য কিছু তুলনা করা যায় না। মানুষের ইমোশনাল একটা বক্স।
Based on this observation, Mendel proposed only
জঙ্গলের
কি বানিয়েছিস
চর্বি খাবি,
না যা
চালাবো
না, আচ্ছা চালা।
আমার না ল্যাপটপ খারাপ হয়ে গেছে এই সুইচ বোর্ডে দেওয়ার পর, সব সময় ক্রিটিক্যাল লো ব্যাটারি, সারাদিন ইউজ করলাম না ক্রিটিক্যাল লো ব্যাটারি, এবার আমি একটা হলুদ কি দেখাচ্ছে ব্যাটারির কাছে, সুইচ বোর্ডটা একটু লেগে যাবে নতুন বোর্ড। আমার ওই ল্যাপটপ কত ব্যাটারি থাকে, সারাদিন ক্রিটিক্যাল লো, অনই হচ্ছে না, মানে হচ্ছে আধ ঘন্টা ইউজ করার পর ক্রিটিক্যাল লো
তো ওটা চার্জ হচ্ছে না,
হচ্ছে, দেখাচ্ছে তো হচ্ছে, এত স্লো রেটে হচ্ছে।
ও তাহলে অন্য বোর্ড চেঞ্জ কর।
दादा सॉरी गाइस, ये जो फर्स्ट आदमी है ना ये मेरा भैया।
Based on this observation, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generation. Now we call them as gene. Gene. Hey, কি দেখছিস? আচ্ছা, টাকা দেখতে হয় লোকে।
সাথে যাচ্ছে
ম্যানার্স জানে না। আর শুনতে পেলাম ধরা পড়িনি বাবা। তাহলে এবার বোঝো, একে যদি কড়া ভাষায় না বলি তো কাকে বলবো?
দাদা আমি
What will be the ratio of progeny when a red colored heterozygous is crossed with a white colored plant? What will the
I know
ratio of
this.
progeny? Oh, phenotype না genotype?
একবার পড় না।
ও একই তো হয়।
জানি তা না কি পড়ি না। Then red color letter is crossed with a white color plant in which red color is dominant and white color. Oh, 1:3.
3
Sorry, is to 1. আমি যা বলছি কেন ভুল হচ্ছে?
1:1 বলছে কেন?
এখানে आंसर একদম তাই না।
What will be the ratio of progeny of a red color heterozygous is crossed with a white colored plant in which red color is dominant on white color?
Hello students, let's read the questions. The question is,
Oh.
what will be the ratio of progeny when a red colored heterozygous is crossed with a white colored plant in which red color is dominant on white color? पूछा गया यहाँ पे दो पेरेंट है, एक क्या है यहाँ पे रेड कलर और दूसरे वाले में क्या है, दूसरा पेरेंट यहाँ पे व्हाइट है। जो रेड वाला हेटरोजाइगस कंडीशन में है एंड व्हाइट वाला जो है यहाँ पे अगर हम देखें तो ये जो है अगर देखें तो अगर ये हेटरोजाइगस कंडीशन में है एंड जो रेड कलर है डोमिनेट ओवर या व्हाइट कलर है, तो अगर कैपिटल आर जो है इसके लिए रेड कलर के लिए एंड स्मॉल आर अगर हम देखें इसके लिए व्हाइट कलर के लिए, तो हमें पता है कि हेटरोजाइगस कंडीशन में कैपिटल आर स्मॉल आर होगा। एंड व्हाइट के लिए अगर हम देखें ये रिसेसिव है, व्हाइट अगर हम देखें रिसेसिव है तो ये क्या होगा, तभी एक्सप्रेस होगा जब भी ये होमोजाइगस कंडीशन में होगा। तो अगर देखा जाए यहाँ पे पेरेंट्स क्या है, पेरेंट्स अगर हम देखें पेरेंट्स का जीनोटाइप ये है ना, जो गैमेट फॉर्म होंगे यहाँ पे, गैमेट्स जो फॉर्म होंगे, गैमेट्स यहाँ पे क्या होगा, कैपिटल आर आ सकता है गैमेट पे या फिर स्मॉल आर आ सकता है। हमें पता है आउट ऑफ 2 एलील यहाँ पे एक ही आएगा एंड यहाँ पे अगर हम देखें दोनों सेम ही है तो स्मॉल आर एक टाइप का ही हो रहा है ना। अगर हम देखिए यहाँ पे F1 प्रोजेनी जो बनेगा, F1 प्रोजेनी जो बनेगा, F1 में दो टाइप आ सकता है, कैपिटल आर के साथ स्मॉल आर आ सकता है एंड स्मॉल आर के साथ स्मॉल आर आ सकता है। तो हमें पता है कि यहाँ पे जो ये वाले होंगे ये क्या होंगे, रेड होंगे, क्योंकि जो कैपिटल आर है डोमिनेट ओवर स्मॉल आर, तो यहाँ पे रेड कलर होगा। ये स्मॉल आर है तो व्हाइट। अगर देखा जाए यहाँ पे रेशियो क्या होगा, 1:1 होगा। करेक्ट आंसर वुड बी द सेकंड वन, 1:1। 3:1 नहीं है, 1:2:1 नहीं है, 9:3।
হার ছিল ভাই।
Allele is said to be dominant if it expressed only in heterozygous. No, it is expressed in both homozygous
সি
and heterozygous condition. It is always expressed homozygous condition এ allele dominant green no এইটা আমার মনে হচ্ছে হবে it is expressed in hetero, এটা মনে হচ্ছে হবে। কারণ হোমোজাইগাসে কেন ওটাকে আমি ডমিন্যান্ট বলবো, তো দুটোই ডমিন্যান্ট। হোমোজাইগাসে তো বিগটি তো ওটা ডমিন্যান্ট কি করে, মানে ওটা তো হওয়ারই কথা। ডমিন্যান্ট তো আমি তখন বলবো যখন নিচু ওর থেকে থাকবে, দেখি করে। হ্যাঁ 3, আচ্ছা 3 ও তো মাথা খাটে না। দ্য গ্রাফিক্যাল রিপ্রেজেন্টেশন যে অফস্প্রিং, এটা তো মিনিট স্কোয়ারই বলে, এটাকে বারবার খালি ঘুরিয়ে ফিরিয়ে জিজ্ঞেস করে, সোজা কোশ্চেন। আরে একটু ক্যালকুলেট।
এটা তো ইজি।
In Mendelian monohybrid phenotype ratio, how many gametes are formed in the F1 generation? How many gametes are formed in F1 generation? গ্যামেট কটা ফর্ম হচ্ছে
করে করে দেখ না, তাড়াতাড়ি আনসার করো।
গ্যামেট তো 4টাই ফর্ম হচ্ছে নাকি?
কেন 2 টাইপস।
2 টাইপস, আচ্ছা। Mendel's principle of segregation mean that germ cell always receive এটা দিয়ে দিই কিছু। One of the paired alleles. If a cross is between AA and AA, small a, the natural F1 progeny will be
AA.
Genotypically AA, phenotypically A. Mendel's first law is law of dominance. The phenotypic ratio of a monohybrid cross RR and small rr in F1 generation is, এটা কি রেড অ্যান্ড হোয়াইট ফ্লাওয়ার নাকি? তাহলে তো এইটা হওয়া উচিত,
কিন্তু সেটা তো বলেনি, জাস্ট এমনি হ্যাঁ দিয়েছে 4:0 তো হবে।
ও
করে দেখে না তাহলে তো আনসার দিতে সুবিধা। করছিস না কেন একটা
মুখে মুখে করছি, মুখে মুখে।
যেগুলো ভুল হচ্ছে কোনো তো কর।
ল অফ সেগ্রেগেশন দেখে নি,
আর নেই কিছু, করে নিলেই হবে।
Hello students, let's read the question.
দেখে নি
Question is
হচ্ছে
the
মাথায় ঢুকে
phenotypic
যাবে,
ratio
আমার
of a
টাইম লাগবে,
monohybrid
আমার
cross,
অনেক কাজ আছে।
capital R and small r in F1 generation is पूछा गया कि जब दो पेरेंट हमने लिए, पेरेंट्स हैं दो, एक कैपिटल R स्मॉल r होगा बस। तो F1 जनरेशन में जब इनका क्रॉस हुआ, यहाँ पे क्रॉस हुआ तो ये क्या फॉर्म करेंगे, एक ही जीनोटाइप आएगा। जितने भी इंडिविजुअल होंगे क्या होंगे, ऑल आर रेड कलर। तो यहाँ का फिनोटाइप रेशियो देखिए 3:1, दो डिफरेंट फ्लावर नहीं आ रहे यहाँ पे, सेम सारे के सारे रेड है टू। और यहाँ जितने भी इंडिविजुअल होंगे वो कितने होंगे, वो कोई भी नंबर होगा। अगर हम देखेंगे तो वो यहाँ जैसे 4:0, अगर 10 इंडिविजुअल है तो 10:0। तो क्या
Law of segregation is applicable for alleles separate from each other during adolescence. Segregation of factor is due to separation of chromosome during missing loss, and is the law of purity of gametes.
হয়ে গেল?
হ্যাঁ 49 পেয়েছি, 7টা ভুল। জিন লো কি, জিন লো কি পড়িয়েছিল দীপু আগের বছর, বলেছি। জিন্স হবে, আমি ভুল করে ফ্যাক্টর দিকে বেড়েছি। এইটা হচ্ছে ঠিক আছে, পরে দেখবো। ডিপিবি 3 আবার 3 করবো 10টা। What would be the color of flower in F1 progeny as a result of a cross between homozygous red and homozygous white? Snapdragon। হ্যাঁ এবার এলো এই যে homozygous F1 progeny. What would be the color of flower F1 progeny? Pink হবে। Incomplete dominance is found in incomplete dominance is found in Mirabilis jalapa। এটা তো আমি পড়িনি।
both
অ্যান্থ্রিয়াম, অ্যান্থ্রিয়াম দেখেছি, ইনকমপ্লিট ডমিনেন্স। In plant red fruit big R is dominant over yellow fruit small r, and the tallness T is dominant over shortness T. If a plant genotype is crossed with a plant RR,
the plant with RR
করে দেখতে হয় বলেই ভাল লাগে না। দেখে একটু মেরে দিই। এটা মনে ছিল, হ্যাঁ 50%. The darkness in plant of F2 generation. Oh, recessive. না রিসেসিভ F2 জেনারেশনে তো
একবার
শো করে, সেটাকে ডমিনেন্ট বলবো, স্মল টি না রিসেসিভ?
একবারই তো শো করে নাকি বোথ আছে, বোথ অপশন আছে?
both of the above. The darkness in plant না both of the above হবে। একটা তো বিগ টি স্মল টি হয়, সেটা তো ডার্কনেস শো করে না।
না বেটে তো থাকে, F2 তে বেটেও জন্মায়, লম্বাও জন্মায়, লম্বা বেশি জন্মায়, বেটে একটা জন্মায়।
হ্যাঁ
তো আমি জানি না, এবার আমার মনে হচ্ছে
দেখি রিসেসিভই বলে ডার্কনেস।
তাও আমরা রিসেসিভই বলবো।
The F2 generation offspring in a plant show incomplete dominance exhibit variable the F2 generation in incomplete dominance. আচ্ছা
কি করলে এটা?
incomplete dominance তো এইটাই phenotype আর genotype same. If two person with AB blood group marry and has sufficient large number of children, this could be either classified as A blood group, AB blood group, B blood group, 1:2:1. Modern technique of protein electro reveal presence of both A and B type of protein in blood group. This is an example. Oh, baba. Oof. The type of gametes formed from 1, 2, 3, 4, এইখানে দেখে নি, আর ওয়াই, আর স্মল ওয়াই, আর বিগ ওয়াই নেই, আর ওয়াই, আর ওয়াই, আর বিগ ওয়াই, আর স্মল ওয়াই, এটা হ্যাঁ এটাই হবে। A gene showing codominance alleles, both alleles expressed in heterozygote. One allele is dominant, allele style in this allele that are recessive to each other. Which of the following genotype does not produce any sugar?
Small I.
Which of the following characteristic represent inheritance of blood group in human? Codominance, multiple allele, 1 2 আর 1 2 আর কি রে, মানে 2 4 2 4 2 4 হ্যাঁ 2 4 5 হ্যাঁ 2 4 5 2 4 5 পলিজেনিক ইনহেরিটেন্স দেখায় না, শুধু 2 and 3 বলছে।
কি आंसर?
ও হো হো ভুলে গেছি রে, কোডোমিনেন্স আর মাল্টিপল অ্যালিল। হিউম্যানে তো কোডোমিনেন্স আইএআইবি স্মল আই, আর মাল্টিপল অ্যালিল মানে দুটোর বেশি আছে, একটা দুটো তিনটে অ্যালিল আছে।
তো আরেকটা হবে না কেন?
2 and 3। এই পলিজেনিক তো অন্য।
কেন অন্য?
ও তো আলাদা কনসেপ্ট, যে একটা জিন যখন আরেকটা ক্যারেক্টারকে এক্সপ্রেস করে, মাল্টিপল কে এক্সপ্রেস করে,
অনেকটা জিন যখন একটা ক্যারেক্টারকে এক্সপ্রেস করে,
ওইটা স্কিন কালার হয়।
ওটা তো হিউম্যানের মধ্যেই হচ্ছে,
ওটা আলাদা। এখানে যেটা বলছে ওটা আলাদা, এই যে প্রেজেন্ট ইনহেরিটেন্স অফ ব্লাড গ্রুপ,
এটা দেখিনি আমি, শুধু হিউম্যান পড়েছি।
25
जिसको जो भी मिलना है सबके साथ।
আজা করব না, আর 3টে ডিপিপি করেছি, 4 5 6 7 কালকে, আর এখন অন্য চ্যাপ্টারে শিফট হয়ে যাব আমরা।
চ্যাপ্টার
সেটা হলো লেখা আছে মরফোলজি।
কি করবি
আগে? মরফোলজির আগে কি করব? প্লান্ট কিংডম করি। মরফোলজি ছোট।
কি করবি? কোশ্চেন
আগে রিডিং, তারপর কোশ্চেন।
রিডিং করতে বলবি?
এই তো বই আনছি তো,
কি বই?
এনসিইআরটি।
এনসিইআরটি থেকে?
এটা আমার এনসিইআরটি সুবিধে। খুলে গেছে। ওকে, ব্রেক টাইম 3 মিনিট পর আমি আসছি কেক ওয়াক করে।
কেক ওয়াক?
কেক ওয়াক মানে এইটাকে বলে, এই যে
কেক দিয়ে ওয়াক।
Just a minute.
I reject this.
क्वालिफाइड है या कंप्लीटली डिजाइन एंड मेड बाय मी। सारा सवाल मैं बनाऊंगा, सारे क्वेश्चन मैं बनाऊंगा, हर तरीके से इसको प्रिपेयर मैं करूंगा।
माना जाता है। हाल में ही इस फिल्म का एक और भी ग्रेट सीक्वल निकल के आया है बॉलीवुड से, जिसका नाम है मैं पाकिस्तान की दीवार।
एकदम से खबर आई कि फवाद आने वाली फिल्म में काम कर रहे लंदन में और ये साथ पानी को
But a recent that it has been confirmed that Hania Amin will set to make her Bollywood debut with none other than Sultan.
जिस देश को आजकल सऊदी अरेबिया, तुर्की और यूएई जैसे एक्चुअल मुस्लिम देश भाव नहीं देते, वहाँ के लोगों के साथ काम करने के लिए बॉलीवुड बना जा रहा है। एक देश जिसकी एडमिनिस्ट्रेशन ने दर्जनों आतंकी अटैक्स करवाए हैं जम्मू एंड कश्मीर एंड द रेस्ट ऑफ इंडिया इन जस्ट द लास्ट फ्यू इयर्स, जिसने इलीगली भारत का एक हिस्सा क्लेम करके रखा हुआ है, उनके साथ बॉलीवुड डेस्परेट है काम करने के। ऐसे लोगों के साथ जो या तो उस एडमिनिस्ट्रेशन को ओपनली सपोर्ट करते हैं या कभी भी उसके बारे में कुछ नेगेटिव नहीं बोलते हैं, इतने सारे आतंकी अटैक्स के बावजूद उसके बाद फिर बॉलीवुड वाले सरप्राइज एक्ट करते हैं कि उनकी इंडस्ट्री मरी क्यों जा रही है।
किधर जाना बेस्ट है, किधर जाना वर्स्ट है, ऑनलाइन भी कहीं हूँ।
So let's start Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker 1969, wherein he suggested that the five kingdom classification was proposed by these Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have been excluded from Plantae, though earlier classification put them in the same kingdom. So the cyanobacteria that also referred to as blue-green algae are not algae anymore. In this chapter we will describe Plantae under algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Let us look at classification within angiosperm to understand some of the concerns that influence the classification systems. The earliest system of classification used only gross superficial morphological characters such as habit, color, name, number and shape of leaves, etc. They were mainly based on vegetative characters or on the androecium structure given by Linnaeus. Okay. Monera, Protista, Fungi, Plantae and Animalia. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have been excluded from Plantae, though earlier classification put them into same kingdom. So fungi and members of Monera and Protista पहले Plantae में रखा जाता था, लेकिन अभी वो प्लांट किंगडम में नहीं रखा जाता है. ठीक है. So the cyanobacteria that are also referred to as blue-green algae, जो blue-green algae बोला जाता है, are not algae anymore. वो अब algae नहीं रहा. In this chapter, we will describe plantae under algae, bryophytes, pteridophytes, gymnosperm and angiosperm. So अब इस chapter में हम लोग डिस्कस करेंगे algae, bryophytes, pteridophytes, gymnosperm और angiosperm को. Let us look at the classification system within the following. The earliest classification system used only gross, superficial morphological characters such as habitat, color, number and shape of leaves, etc. They were mainly based on vegetative characters and on Andresian structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few vegetative characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are much more easily affected by environment. As against this, natural classification system developed which were based on natural affinities among the organism and considered. So artificial system were separated, they separate the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. Artificial system gave equal weightage to sexual characteristics. Artificial system gave equal weightage artificial system artificial system separate the closely related species since they are based on few characteristics. Also, the artificial system gave equal vegetative and sexual characteristics. This is not acceptable since we know that often vegetative characters are based on easily observable characteristics in the environment. As against this, natural classification system developed which were based on natural affinities among the organism and consider not only the external features but also the internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa having a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characters. Numbers and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy that uses the chemical constituent of the plant to resolve confusions are also used by the taxonomist these days. So cytonomy based on cytological character, chemotaxonomy based on chemical constituents. Okay, so this is what the introduction of this chapter. Now we will move in the last part of the chapter, but before that again read the following thing. In the previous chapter we look at the broad classification of living organisms under the system proposed by Whitaker 1969, wherein he suggested the five kingdom classification viz. Monera, Protista, Fungi, Animalia and Plantae In this chapter, we will deal in detail the further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification put them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe Plantae under algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Let us look at the classification within angiosperm to understand some of the functions that influence the classification system. The earlier system of classification used only gross superficial morphological characters such as habit, color, number and shape of leaf. They were mainly based on vegetative characters or on the Andreassian structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characteristic that is not acceptable since we know that often vegetative characters are more easily affected by the environment. As against this, natural classification system developed which were based on natural affinities among the organism and considered not only the external feature but also the internal feature like ultrastructure, anatomy, embryology and phytochemistry. Such classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on the evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to same taxa have a common ancestor. Okay, this is about the things. Now we will read about the most important part, that is pteridophytes and gymnosperm. The pteridophytes include horsetails and fawns. Pteridophytes are used for medicinal purposes and soil binders. So pteridophyte जो include है इसमें horsetail और fawns included है. Pteridophytes are used for medicinal purpose. Pteridophytes यूज किया जाता है medicinal purpose के लिए और ये soil binders भी है. ठीक है. They are also frequently grown as ornamentals. ये frequently ornamentals भी grow होते हैं. ओके. टेरिडोफाइट्स जो है इंक्लूड हॉर्सटेल्स एंड फॉन्स. टेरिडोफाइट्स इंक्लूड हॉर्सटेल्स एंड फॉन्स for medicinal purpose and as soil binders. They also are frequently grown as ornamentals. Evolutionary, they are the first terrestrial plant to possess vascular tissue, that is xylem and phloem. You shall study more about this in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil condition. So first vascular plant tissue possess xylem and phloem. Okay, you may recall that in bryophytes the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophyte the dominant main plant body is sporophyte, which is differentiated into true root, stem and leaf. These organs are well differentiated vascular tissue. The leaves in pteridophytes are small microphylls as in Selaginella or large macrophylls as in ferns. The sporophytes bear sporangia that are subtended by a leaf-like appendages called sporophytes. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophyll. So in Selaginella or large macrophytes as in ferns. So the leaves in Pteridophyta are small microphytes as in Selaginella or large macrophytes as in ferns. The sporophyte bears sporangia that are subtended by a leaf-like appendages called sporophyll. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well differentiated vascular tissue. The leaves in pteridophytes are small macrophylls as in Selaginella or large macrophytes as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyte. In some cases, sporophytes may form distinct compact structures called strobili or cones. Selaginella equistem. The sporangia produce spores by meiosis in spore mother cell. The spore germinate to give rise to inconspicuous small but multicellular. So, leaves the in pteridophyta are microphytes. The leaves in pteridophyta are microphytes as as may in some cases sporophytes may form distinct compact structures strobili or cones. Selaginella equistem. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic phalloid gametophyte called Prothallus. Selaginella equistem, strobila or cones. So 5 minutes of break is coming.
ভালো।
Microphytes, small leaves in pteridophytes, selaginella, large file macro is in fawns. In the sporophyte bears sporangia that are subtended by leaf-like appendages called sporophyte. In some cases, sporophyte may form distinct compact structures called strobili or cones, with selaginella and equistene. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. So, they require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical region. The gametophyte bears small and female sex organ called male and female sex organ called antheridia and archegonia respectively. Water is required for fertilization, transfer of antherozoids, the male gamete released by antheridia, the mouth of the archegonium, fusion of male and gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produce multicellular, well-differentiated sporophyte, which is dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small. Selaginella and Salvinia here called known as heterosporous. Such plants are called, in majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small, are known as heterosporas. The megasporas and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte is the main plant in which retain on the parent sporophyte for variable period. The development of zygote into young embryos takes place within female gametophyte. This event is the precursor to the seed habit. The pteridophytes are further classified into four classes: Psilocida, Psilotum, Lycosida, Lycopodium, Lycosoda, Selaginella, Lycopodium, Spinocida, Equistem, Pterosida, Diopteris, Pteris, Adiantum. So again I will revise to you these Ceobilus, nodes, internode, branch, Equistem. So pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamentals. Evolutionary, they are the first terrestrial plant to pose vascular tissues. Evolutionary, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. We shall study more about this tissue in chapter 6. The pteridophytes require cool, damp, shady places to grow and to may flourish well in sandy soil condition. You may recall that in biophytes the dominant phase of the life cycle of the gametophytic plant body. However, in pteridophytes, the main plant body is the sporophyte, which is differentiated into two root, stem, and leaf. These organs possess well-differentiated vascular tissue. The leaves in pteridophytes are small microphyles as in Selaginella or large macrophyles as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyles. In some cases, sporophyles may form distinct compact structures. अच्छा, so Selaginella, small macrophyls, and ferns are large macrophyls. Selaginella, small microphyls, ferns, macrophyll. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophyles. In some cases, sporophyll may form distinct compact structures called strobili or cones. Selaginella equistem. So distinct compact structure, sporophyll called cones. So sporophyte is a sporophyte bears sporangia that are subtended by leaf-like appendages that is found in Selaginella equistem. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular uh, salvinia forms. Free-living prothallus, gametophytic plant body. Prothallus, the gametophytes require cool, damp, shady places to grow. Because of this species restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. So the gametophyte, which is free-living, mostly photosynthetic, thalloid gametophyte, is prothallus, and gametophyte requires cool, damp, shady places to grow. Because of this specific restricted requirement and need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of antherozoids. Water is required for transfer of antherozoid. The male gamete released from the antheridia to the mouth of the archeogonium. Fusion of male gamete with the egg present in the archeogonium result in the formation of zygote. Zygote thereafter produce a multicellular, well-differentiated sporophyte and pteridophyte of all the spores of kind. Such plants are called homosporous, like Selaginella and Salvinia, who produce two kinds of spores. Macro and micro are known as heterosporous. The mega spores and microspores germinate and give rise to male and female gametophyte respectively. The female gametophyte in the plants retain to their parent sporophyte for variable periods. The development of the zygote into young embryos takes place within the female gametophyte. This event is the precursor to the seed habit, considering an important step in evolution. The pteridophytes are further classified into four classes: Psilocida, Xylotum, Lycosida, Selaginella, Lycopodium, Spinosida, Equistem, and Pterocida, Diapteris, Pteris, Adiantum. That's it. Next is gymnosperm. Gymnosperms are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. Gymno means naked, sperma means seed. Gymno means naked and sperma means seed. The seeds that develop post-fertilization, both the before and after fertilization. The ovules are not enclosed by any ovary and remain exposed. Both are before and after fertilization. Okay? Both before and after fertilization. Okay, I'm taking rest. Okay, so we were reading gymnosperms. In gymnosperms, in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, are naked. Gymnosperm include medium sized tree or tall tree shrubs. One of the gymnosperm, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have a fungal association in the form of mycorrhiza pinus, while in some others, cycas, small specialized roots called coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched cycas or branched pinus cedrus. Okay. Roots in some genera have fungal association in the form of mycorrhiza, that is pinus, while in some others, cycas, small specialized root called coralloid roots are associated with nitrogen fixing bacteria and unbranched cycas and branched pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaf persists for few years. The leaves in gymnosperms are well adapted to withstand extreme temperatures, humidity and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also helps to reduce water loss. So, nitrogen fixing cyanobacteria colloid roots are associated. Roots in some genera have fungal association mycorrhiza pinus. Fungal association, that is mycorrhiza pinus. And cycas smocoroid cycas. The leaves may be simple or compound. Leaves may be simple or compound. The stems are unbranched cycas, branched pinus cedrus. Stems unbranched cycas and branched pinus cedrus. Stems unbranched cycas. Stems branched pinus and cedrus. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also helps to reduce the water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores that are produced within sporangia that are borne on sporophytes, which are arranged spirally along an axis to form lacs or strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into male gametophytic generation, which is highly reduced and is only limited to number of cells. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophyles which are arranged spirally along an axis to form lacs or strobili cones. The strobili bearing microsporophiles and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophyte generation which is highly reduced and is confined to only limited number of cells. The reduced gametophyte is called pollen grain. The development of pollen grain takes place within microsporangia. The cones bearing megasporangia with ovules or mega are called female strobili. The male or female strobili may be born in the same tree, that is pinus, or different tree cedrus. Same tree pinus, different tree cycas. The megaspore mother cell is differentiated into one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovule are borne on megasporophiles which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspore enclosed within the megasporangium nucellus develops into a multicellular female gametophyte that bears 2 or more archeogonia or female sex organ. The multicellular female gametophyte is also retained with the megasporangia. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gametophyte do not have independent free living existence. They remain with the sporangia retained to the sporophyte. The pollen grain is released from the microsporangium. They are carried out in air currents and came in contact with the opening of the ovules born on megasporophyle. The pollen tube carrying the male gametes grow towards archeogonia in the ovules and discharge their contents near the mouth of the archeogonia. Following fertilization, zygote develops into an embryo and the ovule into seed. These seeds are not covered. That's it. The gymnosperm are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop most post fertilization are not covered. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the root called coralloid root, in the form of mycorrhiza pinus, while in some others, cycas, small specialized root called coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched. Cycas, branched pinus cedrus. The leaves may be simple or compound. In cycas, the pinnate leaf persists for few years. The leaves in gymnosperm are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaf reduce the surface area. The thick cuticle and sunken stomata also helps to reduce the water loss. The gymnosperms are heterosporous. They produce spores, haploid microspores and megaspore. The two kinds of spores are produced within the microsporangia that are borne on sporophyles which are arranged spirally along an axis to form lacs or compact strobili or cones. The strobili bearing microsporophiles and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophytic generation which is highly reduced and is confined to only limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophyle with ovules or megasporangia are called macrosporangiate or female strobili. The male or the female cones of the strobili may be born on the same tree, that is pinus, and or in different trees cycas. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucell is protected by envelopes and the composite structure is called an ovule. The ovule are born on the megasporophyle and which may be clustered to form the female cone. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspore enclosed within the megasporangium develops nucleus into multicellular cellular female gametophyte that bears two or more archegonia or female sex organ. The multicellular female gametophyte is also retained within the microsporangium. Unlike bryophytes and pteridophytes, gymnosperm, the male female gametophyte do not have free independent living existence. They remain within the sporangia, retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the sporangia, the ovules, and the microsporophyll. The pollen tube carrying the male gamete towards the archeogonia in the ovules and discharge their currents near the mouth of the archeogonia. Following fertilization, zygote develops in embryo and ovules to seed. These seeds are not covered. Done. কেন? না, আরে সিসিটিভি নেভাবি। Okay. So let's start. Uh, Now we will start about algae. Okay. So algae are chlorophyll bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi. Some of them also occur in association with fungi, lichen, and in animals, sloth bear. The form and size of algae is highly variable. The size ranges from the microscopic unicellular forms like Chlamydomonas to colonial forms like volvox. uh, to the filamentous form, unicellular form, Chlamydomonas. Unicellular form, Chlamydomonas. Two colonial forms, volvox. Two filamentous form, Indothrix. And Spirogyra. A few of marine forms, such as scalps, form massive plant body. The algae reproduce vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, and on germination give rise to new plants. Sexual reproduction takes place through the fusion of two gametes. These gametes can be flagellated and similar in size as in Chlamydomonas, or non-flagellated dissimilar in size, non-flagellated but similar in size Spirogyra. Such reproduction is called isogamous. Flagellated and similar in size Chlamydomonas, non-flagellated non-flagellated similar in size Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in some species. Fusion of two gametes dissimilar in size, that is anisogamous. In some of species of Chlamydomonas is anisogamous. Fusion between one large non-motile static female gamete and a small motile male gamete is termed as oogamous, termed Wallace fucus. Fusion between one flagellated fusion between that's it. Okay. Stripe frond fast. Green algae. Volvox chlamydomonas chara. Laminaria fucus victoria. Porphyry polycythemia. Volvox chlamydomonas chara. Algae are useful in a variety of ways. At least a half of the total carbon dioxide fixation mass is carried out by algae through photosynthesis. Being photosynthesis, they increase the level of dissolved oxygen in the environment, and they are the paramount important source of primary food energy, which form the basis of food. Which are the primary which are the paramount importance as a primary producers of energy rich compound, which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Gelarium and Gracilaria are used to grow microbes and in preparation of ice cream and jellies. Colorella and Spirulina are unicellular algae and are rich in proteins and are used as food supplements even by space traveler. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae, the members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grayish green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and outer layer of pectose. Some algae store food in the form of oil droplets. Vegetative reproduction usually takes place by fragmentation or by the formation of different types of spores. Asexual reproduction is by flagellated zoospores, production and zoospermias. Sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or gamous. The reproduction is a flagellated zoospore. So China में बोलबो उल्टा के Spain चला is the main examples of green algae. Phaeophyci, the members of Phaeophyci or brown algae are found primarily in marine habitat. They show great variation in size and form. The range from marine simple branched filamentous form ectocarpus to profusely branched form as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A and C carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophylл pigment fucosanthin present in them. Food is stored as xanthophyll, chlorophyll A and C, carotenoids and xanthophylls. They vary in color olive green to various shades of depending upon the amount of xanthophyll pigment fucosanthin present in them. Food is stored as complex carbohydrate which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating called algin. The protoplast contains, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and the leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction takes place by biflagellated zoospores and a pear-shaped and a two unequal laterally attached flagella. Sexual reproduction may be anisogamous, isogamous, or oogamous. The gametes are pyriform shaped and bear two laterally attached flagella. The common forms are ectocarpus, dictyota, laminarius, anlagum, and fucus. E.D. Lamborghini Rhodophyci are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where little light penetrates. Red thalli, most of the algae multicellular, some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae uses reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization events. The common members are Polyspania, Porphyra, Gracilaria, Gelidium. Now it's time for biophytes. Biophytes include various mosses and invertebrates that are found commonly growing in moist shaded areas in the hills. Archaeogoniaphore, Antheriophore, Gemmacock, Nizoids. Marchantia, Menthalamus. Funaria, Gametophyte and Bryophytes are also called amphibians of the plant kingdom because this plant can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like, prostrate or erect and are attached to a substratum by unicellular and multicellular rhizoids. They lack true root, stem and leaf. They may possess root-like, leaf-like, stem-like structure. The main plant body of the bryophyte is haploid. It produces gamete, hence it is called gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated antherozoid. The male sex organ called archaeogonium is flask-shaped and produces a single egg. The antherozoids are released into water where they come in contact with archaeogonium. Antherozoid fuses with the egg to produce a zygote. Zygote do not undergo reduction division immediately. The antherozoids are released into water where they come in contact with archaeogonium. And antherozoids were fused with egg to form the zygote. Antheridium and archaeogonium. Antherozoid. They produce biflagellated antherozoid. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division to produce haploid spore. These spores germinate to produce gametophyte. Gametophyte in general importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum or moss provide peat that have long been used as fuel, and because of their capacity to hold water as packaging material for transshipment of living material. Mosses, along with lichen, are the first organism to colonize rock and hence are of great ecological importance. They decompose rock, making the substrate suitable for growth and higher plants. Since moss form dense mat on the soil, they reduce the impact of falling rain and prevent soil erosion. The biophytes are divided into liverworts and mosses. So I am reading again this. Bryophytes are also called amphibians of the plant kingdom because this plant can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophyte is differentiated than that of algae. It is thallus-like, prostate or erect and attached to the substratum by unicellular and multicellular rhizoids. They lack true root, stem and leaf. They may possess root-like, leaf-like or stem-like structures. The main plant body in the bryophyte is haploid. It produces gamete, hence it is called gametophyte. The sex organs in bryophytes are multicellular. The male sex organ in Anthridium, they produce biflagellated antherozoid. The male sex organ are called anthrogonium. Archaeogonium is a flask shape and produces a single egg. Antherozoids are released into water where they come in contact with archaeogonium. Antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spore. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum or moss provide a peat that had been long used as fuel, and because of their capacity to hold water as packaging material for transshipment of living material, moss along with lichens are the first organism to colonize rock and hence are of great ecological importance. They decompose rock, making the substrate suitable for the growth of higher plant. Since moss form the dense mat on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Now we will study liverworts. The liverworts grow usually in moist, shady habitat such as bank of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant of liverwort is thalloid, example Marchantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members are tiny leaf-like appendages into two rows on the stem-like structure. Asexual reproduction liverworts takes place by fragmentation of thalli or by the formation of different specialized structures called gemma. Gemma are green multicellular asexual buds which develop in small receptacles called gemma cups located on the thalli. The gemma become detached from the parent body and germinate to form new individual. During sexual reproduction, male and the female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into foot, seta and capsule. After meiosis, spores are produced in the capsule. These spores germinate to form free-living gametophyte. Next is moss. The predominant of the life cycle of the moss is the gametophyte, which consists of two stages, protonema stage and the leafy stage. Protonema stage, which develops directly from a spore, it is a creeping green branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular brown rhizoids. This stage bears a sex organ. Material reproduction in moss is by fragmentation, budding, and in the secondary protonema. In sexual reproduction, the sex organ Anthridia and Archaeogonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into sporophyte, consisting of a food, set, and capsule. The sporophyte is moss more elaborate than that in liverwort. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of moss are FPS, Funaria polytrichum sphagnum. Spores are formed after meiosis. Zygote develops into sporophyte. So I am reading again the moss. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping green branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender, axis bearing spirally arranged leaves. They are attached to a soil through multicellular and advanced rhizoid. This stage bears the sex organ. Vegetative reproduction in moss is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ, antheridia and arthropodia are produced in the apex of the leafy shoot. After fertilization, the zygote develops into a sporophyte consisting of a food strata and capsule. The spores are more elaborate than in liverwort. The capsule contains spore. Spores are formed after meiosis. The moths have an elaborate mechanism of spore dispersal. Common example of moths are FPS, Funaria polytrichum and sphagnum. Chapter is done. Now the questions is left. Okay. Sure. Now we are doing questions. নম্বর বেড়েছে। ओके, आज के लिए इतना ही। आज हम लोग पढ़े प्रिंसिपल ऑफ इनहेरिटेंस, फुल क्वेश्चन किए और उसका चैप्टर 85% हो गया है। फुल क्वेश्चन इसमें नहीं किए और प्लान किंगडम हम लोग फुल कंप्लीट कर चुके हैं। जो करना था वो मॉर्फोलॉजी रह गया और लिविंग वर्ल्ड, वो हम कल देखते हैं, ठीक है। कल वहीं से शुरू करेंगे, कल शाम 4 बजे 4:15, ठीक है। बाय बाय।